perhaps even more relevant when we look at what happens to place cells when we do the same manipulation. And they actually don't go away. They don't stop firing. And what you just said might be a possible explanation for that. Again, nobody knows for sure that it is the explanation, but maybe
So
place cells are
that
using
is-
that data.
That secondary projection to supplant place cells.
Yeah, so maybe, and this is just speculation because more studies need to be done, but maybe the medial septum projections are more important for the grid cells to do their thing, and the thalamic projections are more important for the place cells to do their thing. Who knows? Yeah. Yes and no. So there is a study where they did that, and basically this was before grid cells had even been discovered, and they did this to see if head direction cells were necessary for place cells. They turned off that part of the anterior thalamus where head direction cells are found, and probably a lot of the theta ones too. And place cells were still okay, actually. So I say, well, that throws out that theory. But this ascending theta projection here is actually quite extensive and complex. It doesn't just include these anterior thalamic nuclei, but also these midline thalamic nuclei. So you actually have a whole set of thalamic structures along the entire extent of the midline thalamus that are doing this, and it's very hard. And in that experiment, they did not get them all turned off. So it's the jury's still out, I would say. Yes. Actually, I think in this diagram, there is one major difference, and it's highlighted in this diagram, which is that the head direction signal goes pretty much only through the anterior thalamus, whereas the theta signal goes through the septum and the anterior thalamus and all these other midline thalamic nuclei as well. So basically, the head direction signal sort of, only arrives in the express lane, whereas the theta signal uses the whole freeway, all lanes of the freeway. Yes?
And when, during class, you said that the head direction cells originate from the dorsal tegmentum Gooden, but then in another slide you said that it originates in the mammillary bodies. And
well, it's sort of so it goes from dorsal tegmental the dorsal tegmental nucleus of Gooden to mammillary bodies, and then to the other. So it's sort of a two-step process.
DTG to mammillary bodies.
Yeah, that's right. And there's reciprocal connections between those two, so it's kind of hard to. There's a bit of a chicken and egg problem there with whether it's actually started in one or the other. So don't worry about that. Other questions? Okay. Grid and place cells on a linear track. This is what we started the lab with, and I think the rest of this stuff in here is basically what I used. We're bleeding into lab 3, which is fine, so I'll keep going through that. So this is getting to your earlier question, where basically we said that a grid cell may be detecting synchrony among like 2 oscillations, whereas a place cell is detecting synchrony among many. And you can see here in this diagram how all the peaks of the oscillators line up at this spot where the cell fires, so they become synchronized there. But nowhere else do you see all of them lining up like that. So this just reiterates this point that placing grid cells may be very much the same animal, but one is detecting synchrony among a few rhythms and the other among many. So we just did this as well. So when we turn off the medial septum, grid cells go away, but place cells don't. Here we see spatial coding in 2 dimensions, which is moving out of this simple linear track examples that we've been talking about, where theta oscillators become synchronized at certain points on a line, and starting to think about how is it that we can make this process work. How can place and grid cells fire at a single location in a 2-dimensional environment? And what I said was, and this was then sort of part of the theme of the 3rd lab, is that we just simply have to assume that the theta frequencies depend upon velocity in 2 dimensions instead of 1. And in one dimension, velocity is basically speed. There's also direction because you can go left or right, but there's really one major component of velocity, which is speed. But in 2 dimensions, you have both speed and direction. There's 2 dimensions of velocity in 2-dimensional space. And those 2 dimensions can be separated out as speed and velocity. I mean, speed and direction. So we basically then have to imagine that the frequency of a theta oscillator should vary not just with speed, but also with direction. And if that's true, then detecting synchrony among those kinds of theta rhythms would give us spatially tuned neurons in 2 dimensions. In other words, rhythms that are modulated by both speed and direction synchronize with one another at specific locations in 2-dimensional space. So a neuron that detects synchrony among rhythms like that will fire at certain locations in 2-dimensional space, as grid and play cells do. Does that make sense? That's as deeply as you need to understand it. So if that does make sense, don't worry about it. Yes. Rhythms that become, rhythm generators that become synchronized, rhythm generators that have frequencies which depend on speed and direction will get synchronized with each other only at specific locations in 2-dimensional space. So any synchrony detector that detects synchrony among such rhythms will fire only at specific locations in 2-dimensional space, as place and grid cells do. So that's clear?
What was the last part that you said?
As place and grid cells do. So any synchrony detector that detects synchrony among such rhythms, i.e. rhythms that have frequencies depending on speed and direction, will fire only at specific locations in 2-dimensional space. Yes?
When the rat is on a linear track, do we still see the hexagonal pattern of grid cells?
When a rat is on a linear track, does it still have to go in a pattern of grid cells? I'm not sure what you mean by that.
So let's say the rat is running on a track, and then you're recording from a grid cell. So I'm asking if you still see those hexagonal pattern if you're going to see it from a grid cell.
Well, that hexagonal pattern is a 2-dimensional pattern.
Okay, so
So you cannot draw a hexagon on a line, right? So the best you can see is something like this, a 1-dimensional cross-section through a 2-dimensional hexagonal pattern, which is what this looks like. Yes?
For that statement, rhythm generators have frequencies that depend on speed and direction. Is it speed or direction?
It's speed and direction.
Speed and direction?
Yeah.
So both?
For both, it's both, yeah.
Okay, so grid and play cells both depend on both speed and direction.
Well, again, this is where it becomes useful to keep track of the distinction between what I'm calling rhythm generators and what I'm calling synchrony detectors. Theta cells are rhythm generators. They are the things that produce the oscillations that get synchronized with each other. Grid and play cells are the synchrony detectors. They are the things that fire when they detect synchronization among the rhythm generators. So what you just said is correct, but substitute theta cells for grid cells.
Right.
Okay? Okay. Okay. Other questions? Okay. What we spent the last part of the lab on week 3, this week that we just finished up doing, was doing an analysis to sort of test this result, which was recently came from our lab, and we were able to see some cells that appeared to do this. But this is the direction part, so where we said that if the theta frequencies need to be modulated by direction, then are they? And the way we tested that, and we actually did it for ourselves in the lab this week, was to take the theta cells and look at their burst frequencies during movement in different directions. And again, I'll emphasize something that I said in response to an earlier question, which is that we are now talking about the direction in which the rat is traveling, not the direction in which the head is facing, which is what we mean by directions for head direction cells. And that is emphasized by this diagram in which you see us yanking out little segments of the rat's path during which it was moving in different directions. And we ask, what was the frequency of the theta rhythm during those movements in those different directions, and was it different? Did frequency depend on direction, as the theory predicts? And the answer is yes, it does. And we were able to take an autocorrelogram and a power spectrum of the theta bursting during movements in each of these 8 directions. So these are the different directions, and these are the autocorrelograms and power spectra. And from the power spectra, we can measure the burst frequency, as we've discussed already, and we can plot that on the y-axis of this graph. How does the burst frequency vary with movement direction? And when we average over a number of cells, we see that it appears to vary in this very nice way of being highest burst frequency in a specific direction, the preferred direction we call it, and lowest in what we'll call the anti-preferred direction. So this is exactly what we would need to make this whole system work. So it's fun to see that it might be there. There's, yes?
So that theta cell that you showed us in your lab down below, so like at that time that I saw it, was in a particular corner, it was like the top right corner, so we're saying that it's not only dependent on that it has to go to that spot, but it also depends on how it got to that spot, so.
For the theta, you mean?
Yeah, for the direction dependency that you're talking about, it depends on how the rat arrived at that spot.
Well, so the burst frequency of the theta cell varies from moment to moment depending upon the speed and direction in which the rat is moving from moment to moment. So again, let's keep the sort of the rhythm generators and the synchrony detectors separate from one another, because it's the synchrony detectors that do things at specific locations, like up in that corner that you're talking about. They fire when the rat is in a specific location because synchrony among theta rhythms occurs only at certain locations. It is the rhythm generators that do things that depend upon the rat's momentary speed or direction. And so what the theta cell does when you listen to it down in the lab doesn't really seem to depend at all on where the animal is or how it got there, as you're sort of saying. What it depends on is in what direction and at what speed the rat is moving at this moment, regardless of where it is. Does that make sense? That's the short answer to yours. There's just an important. It breaks down into neurons that encode position, which would be our place and grid cells, versus neurons that encode velocity, which would be our rhythm generators, our theta cells. And position and velocity are related to one another in a specific way. Velocity is the time period of position, but we won't get into that too much. So, yes?
In the power spectrum of those autocorrelograms, I don't see how they can produce those different power spectrums. It seems like all those autocorrelograms are the same thing.
Yeah, well, so you don't see why the power spectrum looks different for these, right?
Yeah.
Well, so that's a good question. So the peak of this one looks like it's maybe at about 8. And the peak of this one looks like it's maybe at about 8.3, okay? So that means that the difference between this and this is that if you count the bumps, there's 8 here and 8.3 here per second.
Oh, so it's part of this.
So your eyes, again, this is where we're using the power spectrum to show us something that our eyes can't really see in the data. And this is only, this is not 1 second, this is actually only 0.8 seconds in this case. So